For thousands of years, humans have looked up into the sky and dreamt of exploring what lies beyond. Now, this is becoming a reality. By 2030, NASA aims to send a manned mission to Mars and essentially take our first steps on the red planet. While rocket technology is advanced enough to take us there, we are still uncovering new things about how space can affect human health and what challenges astronauts will face in long-term space flight. The highest concern right now is the bugs that all of us carry on us. Bacteria cause a lot of diseases. In previous studies that NASA has done, they've found that bacteria move faster and can potentially cause more disease in space than they do on Earth. Our project is looking at how and why these changes happen in hopes that we can potentially mitigate these effects in the future. In biology, everything moves from DNA to RNA to protein. While genes in DNA are constant, their expression in RNA and the levels of RNA expressed is dependent on the environment that the bacteria is in. Different environments turn on different genes, which means that different RNAs will be in the bacteria. To test this, we will be sending up a capsule to the International Space Station that has three separate chambers. The first chamber will include inactivated bacterial spores. In the chamber adjacent to that, we will have liquid broth, which is essentially nutrients and water that the bacteria can use to grow and divide in space. In the chamber next to that, we will have a chemical reagent that preserves RNA. 5 days before the experiment is closed up and sent back to us, we will remove the first chamber, flooding the bacterial spores with the liquid broth that has the nutrients in them. And we will give the bacteria 3 days to grow, divide, multiply in space, and have the weightlessness change the RNA that they make. 3 days later, we will release the last chamber, flooding this mixture with the chemical reagent and essentially preserving the RNA within the bacteria.
After a month and a half on board the International Space Station, our experiments will undock and parachute back down to Earth, where we'll begin our analysis and processing. That capsule that contains preserved bacteria is actually a sample of preserved bacterial RNA, that crucial intermediary molecule which regulates bacterial behavior and structure. We can dissect the RNA to understand how bacteria are becoming more pathogenic and regulating their pathogenicity in space. To do so, we'll use a technique known as RNA sequencing. In RNA sequencing, RNA is converted to a stable intermediary known as cDNA, which will then be fed in to a high-throughput Illumina sequencer right here on campus. The sequencer will read small fragments of RNA, generating 150 million short sequences or 7.5 billion bases from each individual sample. But how does this help us understand how bacteria become more pathogenic? Well, each of these small fragments actually belongs to a fragment of RNA, which, using the computational power of Maryland's Deep Thought 2 supercomputer cluster, we can map back to genes from the bacterial genome. Thanks to years of work and painstaking research by graduate students across the world, we know the names and the functions of each of these genes. By quantifying how frequently we see each gene appear, we can understand how bacteria are turning them on and off and regulating the various functions they affect. By profiling genes that are differentially expressed between our Earth and space arms, we can isolate genes that respond to microgravity and that may hold the key to understanding how and why bacteria become better pathogens. In turn, these genes become targets for candidate drugs and next-generation therapies, which will allow astronauts to stay safe and travel in space for longer durations. Other benefits of this experiment include the fact that RNA sequencing not only profiles the RNAs we're interested in, but also every other RNA in the cell, and can be used as a publicly accessible and queryable database for scientists with questions about responses to microgravity by bacteria at any point and any time. Our timeline is a launch in February 2017, followed by return to Earth in April. Our analysis will run from April to May, and from June to July we hope to be preparing our results for publication. We hope that you'll join us as we seek to understand the limits of human health and push the boundaries of human-manned space exploration. Thank you from all of us at the University of Maryland.